Okay, so there will be twice as many of you on Wednesday. Yes, probably. But before that, we have to have, and it's only one clear question, so we'll have one, and then everyone who needs to go and study some more can leave. The live attenuated influenza vaccine for influenza A, H1N1 for 2015-2016 contained how many wild-type segments? And you can read the numbers. What does it mean by quote-unquote wild type? Problem with wild type is you've got so much variation, of course, from year to year. So what do you call wild type? So, yeah. Wild type's supposed to be a really complicated thing as far as viruses are concerned. So. Or late Sunday night, as the case may be. 4, 3, quick vote. And yes, it is 2 segments. Which 2 segments are they? Which genes would they code for? H, A, and NA. So those are the 2 segments in the live attenuated influenza vaccine, also known as flu mist. So it's the reassortment, which happens in the test tube, that's putting those together. So the other 6 are all the attenuated form, the cold-adapted attenuated form of the virus. Yeah, Luke.
So it's not 2 complete virus strains that are in there, as in, like, I thought it was seasonal, 2 of the seasonal flu strains, but you're saying it's just an HA and an NA?
It's just those 2 segments, and so maybe I didn't do a good job of explaining that, sorry, but yes, just those 2 segments that are taken and then put into that strain, which is one of the reasons I think it's such a cool technology and why whenever I have the chance I take flu mist instead. There are other reasons too, but that's a different story, yeah.
What is the geographic scope of the annual decision of what goes in the flu vaccine? Is it U.S. only, or is there global coordination? Or?
Okay, yeah. So the question here is basically who decides, and it's the WHO that makes the recommendation, and then it's each individual country that decides what to do. I would say 99.99% of the time they take the WHO's recommendations. I don't know of any cases where they haven't, but it's certainly possible. It's up to the individual countries. So the WHO has no actual powers to do that. WHO has all kinds of other issues, but again, not something that we need to get into as far as this course is concerned. Okay, so way back when, after the last midterm, we started with Lambda. And I will go through all this stuff relatively quickly. Please stop me, slow me down as I'm going through all of these things. So, and actually, let's hide this guy. So we don't need to see that anymore. So, as far as Lambda is concerned, of course, we've got lytic, we've got lysogenic growth. In terms of lytic growth, it's really all about these 2 promoters, PL and PR. And the main thing with PL is it expresses the N protein, which is a transcriptional anti-terminator. As soon as you have that anti-termination, you go this way from PL, which is not as critical. Much more important is from PR. You go through and then catch up with Q, which is the second anti-terminator. And once you express Q, that allows expression of all of these genes over on this right-hand side, which includes all of them over here on the other side, because these are circular genomes. And so that's what happens when you have lytic growth. It's expression of N followed by expression of Q. Another quick thing to take a look at here is that we are also expressing, even in the absence of N, we're expressing lots of CRO. On the other hand, in lysogenic growth, we're expressing lots of our friend C1, also known as the lambda repressor. As soon as you express C1, you shut down expression of PR. Do the same thing, shutting down expression of PL, but what's important here is looking specifically at PR. How does it happen? It's because the lambda repressor, like its name, mentions, is repressing transcription from the PR promoter, but there's a positive feedback loop which leads to extra expression of the C1 protein. So it's a positive feedback loop. So it's all about the relative concentrations of CRO and C1. And someone asked me about this after lecture the other day. It's really all about these concentrations right here, the low concentrations of C1 and low concentrations of CRO. So the low concentration of C1, it's bound to these two operators, R1 and R2, and L1 and L2, which activates the promoter for repressor maintenance and turns off PR in the case of low C1. In the case of low CRO, you have occupancy of OR3, which means that PRM is off, no occupancy of OR1 and OR2, which means that PR is on. So this is going to be lytic, this is going to be lysogenic. Happy? Yes? No? Huh? That was like forever ago. I've forgotten it all. I haven't studied it yet. So the main question here, of course, is how do you make that decision? Do we go lytic or lysogenic? And the important thing here really has to do with the concentration of C1. But how do you get the concentration of C1 in the first place? That depends on the concentration of C2. C2 depends on C3. But C2 is really that critical point because C2 is unstable. C2 would normally lead to expression of C1. How do you know that? Because if you have a mutation in C2, that means a clear plaque, just like you'd have with a clear plaque if you have a mutation in the lambda repressor. And so it's the expression and stabilization of C2 that leads to your lysogenic growth. What else happens is you have integration, and I think I didn't do a terribly good job of explaining integration last time we talked about this, although in that other course that I taught last term, we went over this probably ad nauseum. The integrase gene is a tyrosine recombinase. It's absolutely critical not only for putting the phage genome into the host genome, but also taking it back out. It's a classic enzyme. All it does is catalyze a reaction, and those reactions are cutting and joining of phosphodiester bonds. What happens is the integrase binds to the attachment site, ATT, either on the phage genome, which is P, or in the bacterial genome, which is B. So at P and at B. Those are two DNA sequences that the integrase is going to bind to. In the integration reaction, which is shown right here, you have just the integrase protein and the integration host factor. Again, not surprisingly, this is a host gene, this is a bacterial gene. All it does is bend the DNA. And then with just IHF, you have integration. And so what happens is you have a crossing over that takes place here. This is the P, one of the flanking sequences in the phage genome that crosses over. So you end up with P next to B prime. So just literally following the X here. The other side, B following over to P prime. That's what you have over here. Once you're integrated into the host genome, you have a left and a right, because, of course, it's all in the genome now. You don't have anything left. This integration is in a very specific place in the genome. It's right between the galactose operon and the biotin operon in E. coli. This is a very specific interaction. How do you get that specificity? It's due to the sequence, which is present here in O. And this is the operator site for integration. This sequence is identical in the phage genome and in the host genome. These other sequences, P and P prime, B and B prime, are slightly different between the two. In the presence of excisionase, which is only made if you're going down the lytic cycle, then you have reversal of this reaction, you end up with a circular genome. So I'm going to spend a little more time talking about integration. Is this clear, unclear? How's he going to ask questions about this? The answer is C, or not, as the case may be. Okay, how many people had a chance to check out the video? Anybody? Should I have bothered to put it on reserve in the library? Did anybody try and find it and it wasn't there? Does anybody look at it in the next 48 hours? Yeah. Okay, so I won't take it off reserve. Part of the reason I ask is because, I know some of you stuck around a little bit after lecture last time, saw what I think is a really nice animation of how you get packaging that takes place. And it really has to do with this terminase enzyme sticking at the end of your concatenated genome. So you've got multiple copies of the genome. Why do you have multiple copies of the genome hooked up end to end? Because replication is happening in a rolling circle process, exactly. So you end up with multiple copies all attached end to end. The terminase protein binds to one of those ends, packages one copy of the genome inside a prohead structure, and then cuts this off, finds another prohead, puts it into the next COS site, etc. So that's the whole process on how you package the genome of lambda inside the head structure. And there's again, shameless plug for my Facebook page. There's a really interesting biophysical analysis of what the DNA looks like when it's packaged inside some of these phage heads. Not, in this case, not just lambda, another related bacteriophage, but it's this interesting sort of sand kind of state, which is sort of somewhere between solid and fluid. Yeah?
Does the terminase go into the virion head, or does it go to the next concatemer?
So, no. So the terminase stays outside the head. So it's a non-structural protein. It's just one of those that's made at one of the very late genes, but it's just involved in packaging. It's not part of your final virion. Okay, other questions on lambda? No, I don't have the clicker question, but there'll be some on Wednesday, I promise. So switching gears, talking a little bit about the now all RNA viruses from now on to the end of the lecture, all RNA viruses all the time. These are the plant viruses, and just one example of literally thousands of different plant viruses. Important here is the transmission of these plant viruses. Many of them, like cucumber mosaic virus, which is the main one that we talked about, they need multiple genomes to get inside the cell. Getting inside the cell is really, really hard, so it's great to have a vector, like our friend the aphid here, who has lots and lots of virions attached to this mandible where it's making a hole in the cell. And why do you need multiple capsids? Because you need multiple genomes. Each capsid encodes one of the genome segments. So this is one of the really bizarre things about a number of these plant viruses, is that each of the genome segments, unlike the case for influenza, where they're all inside one handy-dandy envelope, one virion, they're in multiple different virions. But because of this infection process, this is clearly a way that they're functional. In fact, cucumber mosaic virus is one of the most successful viruses out there. So here, virions themselves package about 3,000 nucleotides, and this is one of those sets of 3,000 nucleotides. Here's a second one, and then these two go together. 2 + 1 = 3. It's kind of math we like for this kind of class. So the RNAs that are here, not surprisingly, encode RNA-dependent RNA polymerases, capping enzymes, etc. So they make the caps. So it's the viral proteins which are making the caps on here. And then your structural proteins, which you have down here. The other really unique aspect of a lot of these plant virus genomes is what's going on down here at the 3' end, and that's the tRNA-like structure right at the very 3' end of each of these genome segments. And that particular structure seems to be mostly important for protecting the end of the genome from getting degraded. As, again, that last term, that last course, what you have at the 3' end of your genome is really important because that's where you have lots of exonucleases that are going to be chewing in at any kinds of ends they can find. And you've got some kind of secondary structure that can block it. If, in this case, and in some of these plant virus genomes, you actually have an amino acid which gets attached to the 3' end of that tRNA-like structure, that amino acid at the end is something an exonuclease can't deal with. So it completely protects that particular end of the genome.
And then that's that AAT RNA synthetase?
The aminoacyl tRNA synthetase, yes.
That's what adds this.
So it's aminoacyl tRNA synthetase, which would normally be adding amino acids to tRNAs. It binds to, recognizes the structure, and adds an amino acid to it. Yeah?
Are all the genomes present in the same concentrations? So you have, you know what I'm saying? Like there's RNA1, RNA2, and RNA3, they're all the same amounts.
Yeah, so the question here basically has to do with relative amounts of each of those different genome segments. And the answer is I don't know, which means it wouldn't be on an exam. But presumably you have to then, when you're packaging, in order to get the appropriate amount, you're going to have equivalent amounts of each of those genomes, or just really massive amounts of each of those RNAs. And it's probably the latter, that you've got so many of each of these RNAs that just by chance, and the fact that each of those capsids packages almost exactly 3,000 nucleotides, is that you'll have enough of the capsids with enough of those different genomes that will end up functioning. But it doesn't seem to be any particular regulation of replication of one more than another. Okay, so I mentioned some of these genes already. Again, methyltransferase, you have to have something which is forming the cap on your messenger RNA. It needs to get translated. How do you get translation taking place? Well, you've got those picomaviruses that we'll talk about in a second, and the flaviviruses, but for most part, you're using cellular translation. All viruses need cellular translation. How do they recognize the messenger RNA? Through that cap structure. If you're replicating as an RNA in the cytoplasm, where are you getting caps from? You've got to make your own. And so this is one of those proteins here. The protein expressed by RNA1. I wouldn't remember the numbers here because I can't remember them either. Also, of course, it's an RNA-dependent RNA polymerase. And then the other thing which is specific to these plant viruses are these movement proteins, which are really important for getting the genome from one cell to the next cell and getting systematic infection in the whole plant. Because if you're dependent on having some kind of vector, so the aphid's going to feed on the plant, having that vector come back and feed in exactly the same cell that it fed on in the first place is really, really, really unlikely. So you have to have some way as the virus for spreading throughout the whole plant. So when the next aphid comes along and eats, then it's also going to get lots of virions associated with its mandibles and be able to move to the next place. And so that's just a reminder here. These movement proteins are getting the virus genomes. Apparently, it's just the virus genomes which are being moved, not actual virus capsids. The virus genomes which are being moved from place to place inside the plant, either long distance through the vascular system of the plants, or just from cell to cell through these plasmidesmata processes. And then potentially actually I was going to call it the coolest virus, but it's the wrong terminology, or the hottest virus. Not quite. We'll talk about the best ones at the end of the term. But that we talked about so far is the Curvularia thermal tolerance virus, which infects the endophytic fungus, which lives in the roots of these grasses and allows them to survive at these very high temperatures. So without the fungus, without the virus, we don't get any survival at these high temperatures. And it's not really a plant virus, I know, but close enough. Yeah, Charlotte.
My first question is this segmented genome tactic pretty common in plant viruses? A, that's my first question. That's
Yes.
sort of okay, thank you. And then B, these movement proteins, do those at all also participate in these, because I would imagine that if you have some distance between, RNA1 and RNA4, that maybe these movement proteins also allow all the genomes to be in the same place at the same time, or again, is it more of a concentration thing?
So again, the first question is, how common are that these plant viruses have segmented genomes? Quite common, but by no means the only case. And tobacco mosaic virus is a nice example of just a single segment. So there are a number of those, but I think absolute number-wise, probably a little more in terms of the segmented genomes. But again, I'd have to go back and check those numbers. Second question was having to do with movement proteins and whether they bring genomes together in order to make sure you get what we were talking about before, making sure you've got all of the appropriate genome segments that you need in order to make a functional infection. And I think the answer to that is, well, first, I don't know. And second, lots of movement proteins are doing lots of different things. And so the fact you have movement proteins is really important, but how they function seems to be different for lots of different virus types. So it's probably one of the most active areas of research in looking at plant viruses.
You said for the test that you weren't going to ask us, say, is 1A a methyltransferase or anything like that. So we just need to know that plant viruses need a methyltransferase in their immediate early genes, for instance, for purposes.
Mm-hmm. Yeah, for purposes of this. So the terminology gets really overwhelming, as I'm sure any of you have been trying to study for this exam have noticed. And so there'll be a few things that I'll talk about a whole bunch, like Ns and NPs, or HAs and NAs. Okay, I think those are reasonable. RNA1, protein 1A, 1C, I don't think that's reasonable. And if you're wondering about that, feel free to send me an email, and I'll tell you to remember everything. No. I'll try and be good about that, and also as going through here, some of these things, like, NS. I can't remember what NSP4 is for flaviviruses, which is different for different flaviviruses. I don't think it's reasonable for me to expect you to remember that. This is not a med school class. Oh, no. Yeah, so TMV. Can I edit that out of the recording? No. So just getting back to Trevor's question about this. So TMV is one of those plant viruses for which there is only a single genome segment. And this is quite different than the other RNA viruses, which we'll talk about in the next couple of minutes, whereby the nucleocapsid protein, which is also known as the coat protein for TMV, is really sitting on the outside of the RNA. So the RNA is here in red, and then the helical structure along the outside here is protecting that RNA. And again, not surprisingly, because these are naked virions, they're present in the absence of an envelope. So any more questions on these plant viruses? And again, go to OSU. I think there are like 4 courses on plant viruses that you can spend terms working on. Fascinating, critical, wonderful. Yes, good. Let's move on. Okay, so now those viruses that infect human primates as opposed to non-human primates. And also a bunch of other things. So the picoRNA viruses, I try not to say you got me. So yeah, I try never to say that particular pronunciation of it because I think it's ridiculous. But the picoRNA viruses, there are lots and lots and lots of them. And I think that was the main point of this slide, is mostly what we're talking about in this class and other classes is polioviruses, but the rhinoviruses are much more common than these polioviruses, and in fact, I've got a little bit of probably a little rhinovirus infection going on right now. The other thing here is that hepatitis A virus, again, really good description of the fact that hepatitis can be caused by a whole bunch of different virus classes. It's not just one versus another. And then there are probably way, way, way more picoRNA viruses out in the environment that have nothing to do with infections of animals, be they humans or otherwise. And that's one of the, I think, really open areas in terms of trying to understand how a lot of these viruses function, circulate, evolve, etc. We have talked about this. In fact, I just went back and checked yesterday. We're now up to 13 wild polio infections this year, all still in Pakistan and Afghanistan. In fact, the last two were also in Pakistan. So almost gone. And in fact, if you go to the polio eradication site, the main thing they're talking about is removing type 2 polio from the oral polio vaccine so that you don't have these vaccine-derived polio cases. So at least for type 2 polio, that's no longer in the oral polio vaccine. So there shouldn't be any more of this reversion which takes place where you end up back with a virulent strain which actually can cause paralytic disease. Much more importantly, as far as these picoRNA viruses are concerned, is how they function. How you getting inside the cell. Of course, these genomes have a protein bound to the 5' end of the genome. Why? Because it's the primer for replication. So VPG also seems to be really important for getting the genome inside the cell. Poliovirus and most of these picoRNA viruses bind to receptors on the outside of the cell, and then there's a big conformational change that happens in both the VP1 and VP4 proteins, which basically pulls the genome inside the cell through a pore complex. So this is extremely similar to what happens with a lot of the bacteriophage, that we have a conformational change on receptor binding that pulls your genome inside the cell. No, there's not transcription that's happening here and pulling the rest of it in like T7, but this is the process whereby almost all of the picoRNA viruses are getting their genomes inside the cell. That genome, again, it's got a protein bound to the 5' end, one ginormous open reading frame in the middle, this polyprotein that then gets chopped up by the various different viral proteases in terms of getting you all of the individual pieces that you need to make your virion, this is all of your capsid proteins here, and then all of your non-structural proteins which are involved in replicating the genome and or putting these capsids together, etc. The poly A tail here is made how? So I heard stuttering, which is incorrect. It's encoded for in the genome. So poly A tail here is a genome-encoded segment. So actually these picoRNA viruses, at least the poliovirus, some of the other ones are actually kind of different. But for poliovirus, it's actually encoded for in the genome. So it takes a couple hundred nucleotides, which are either going to be U in your antigenome or A in your genome sequence. So it's quite a bit of the genome which they use in order to encode these poly A tails. On the other hand, they don't have caps. What do they use instead? Internal ribosome entry sites. There was some question here about which protease is involved in cutting the EIF4G structure, so the host translational initiation. It's actually the 2A protein in polio. I may have misspoken. I probably said 3C in the past. Turns out it is 3C for some other viruses, which again is this whole confusion business about which one is which. So it's a viral protease which cuts that cellular translational machinery. And we'll just stick with viral protease instead of trying to have those individual letters and numbers here. At least I'll try and do that when I revise the exam and proofread it in the copious free time that I have between actually finishing writing it, which is not done yet, and Wednesday morning. But here, again, it's the internal ribosome entry site, which is critical for getting your small subunit of the ribosome to the start codon for the polyprotein. And once you have the small subunit of the ribosome, your initiator tRNA, then the large subunit of the ribosome will come in and you'll translate the whole rest of the protein. And so that's how it works for the picoRNA viruses, also all the flaviviruses. Dr. Hirsch didn't talk too much about that, but most of the flaviviruses also have these iris structures, I should say, rather than sequences, present at the 5' end of the genome, which is really important for getting translation to take place. On the other hand, you also need to replicate the genome of these viruses, and again, always the issue when you've got an RNA virus, making messenger RNA versus making first anti-genome and then genome sequence, which you're going to end up packaging. How does that work? Of course, it's that VPG protein, which is serving as the primer. VPG is also known as 3B. Yes, I expect you to remember VPG. No, I don't expect you to remember that it's 3B. That is associated with membrane vesicles. True for many, many, many of these RNA viruses replicate through. See, I got so excited about replication, and so I'm trying to get all the way down to the 3' end, so I can replicate that. So here, the VPG gets 2 uracil residues added to it, and that process happens through this Cre element, which is shown here as being in the middle of the genome. It's actually quite close to the 5' end of the genome. So VPG interacts with 3CD, which is the polymerase, particularly the 3D protein, which will then put a couple of U residues onto VPG. Once it's got a couple of U residues on it, it's obvious what those can bind to. It's the poly A tail at the 3' end of the genome, over here where I'm falling over the chair. And that then allows you to extend all the way back through the rest of the genome. How do you avoid having translation coming the other way and banging into the replicase? That's through the cellular poly-C binding protein, the 3CD protein, also binding to secondary structures present here at the 5' end of the genome. You're not going to have the 3CD protein made until you've already translated your whole polyprotein. And so this is a nice way to control for having already translated your genome. 3CD can come and bind here at the 5' end, which will block translation taking place and allow the replication to take place, making your negative strand. On the other hand, once you've made the negative strand, now you don't need to worry about the ribosome, because the ribosome and the RNA-dependent RNA polymerase are going in the same direction. And so you can end up with multiple copies of your positive strand from a few copies of your negative strand of the genome. Yes, happy, wonderful. We could all draw it out on the exam, which would be great if I actually had time to grade all of them, which I don't. So there'll be some kind of question about this on there. Any great ideas on multiple choice questions? I always try to come up with good multiple choice questions for replicating picoRNA virus genomes, and it's a pain. So I could just ignore it, right? Not even bother with the question. That would be too easy. Okay, so the other thing I wanted to mention, I think I skipped over this also probably far too rapidly at the end of lecture when we were talking about the picoRNA viruses, are the rhinovirus genomes. They all have irises, but their actual structures, particularly at the 3' end, are really quite different. And so almost all of these genomes is about 100 genomes that were sequenced. They didn't have poly A tails, they had hairpin structures. And the hairpin structure is just self-complementary piece of RNA, which also appears to be important for protecting the 3' end of the genome from being degraded. And so that was the thing that I wanted to mention there. Any questions then on the picoRNA viruses? So now we wrap an envelope around them and they become flaviviruses. So these are a couple of slides from Dr. Hirsch's lecture that I wanted to emphasize here. Yes, there will be some questions on there. He's given me 2. I've made up a few more. So a couple things about general flaviviruses, and in particular yellow fever, because this was by far and away one of the best studied here. And the really, I think, fascinating aspect about this, and what Walter Reed is given credit for, and whatever his poor assistant was who ended up dying of yellow fever, I forget. But that the virus actually needs to be in the mosquito for up to 12 days before you can get more transmission. So for yellow fever, for dengue, for Zika probably as well, not for hepatitis C, the virus needs to be in the mosquito and replicating in the mosquito. So it's an absolutely critical step. You can't get yellow fever from another infected person if there are no mosquitoes around. You can't get dengue from another infected person unless there are the mosquitoes around. And so it's that replication process which is really critical. And because mosquitoes are so critical for this, people tried, and these are those totalitarian regimes, to get rid of mosquitoes, particularly the Aedes mosquito, and they did a really good job until 1970. And then they sort of slipped a little bit. Democracy, yuck. So this is the case that we have basically now. Aedes aegypti is all over the place. And of course, being all over the place is able to transmit this disease because it can pick up those viruses, be in the mosquito for up to 12 days, and then have another infection process that takes place. So not surprisingly, where you see dengue infection is basically where you see Aedes infection. Pretty much all throughout the world, all in the tropics, and it's probably a lot of these cases here is underreporting for a lot of this. Unfortunately, many of these people, when they get infected, don't have much in the way of symptoms. It's only extreme symptoms which are the ones that get reported. And so probably that's what's going on, particularly in places here, like the Central African Republic, where they're not reporting any particular dengue disease. Turns out that I was, in fact, incorrect in my clicker question, so I'll be going back and correcting that. There aren't actually that many people who die from dengue in any given year. It's only about 25,000 or so worldwide. And how many people die in the U.S. from influenza every year? 30,000 or so. So influenza is way worse than dengue or all of these other things. And people dying from Zika, very, very few. At most, 5 or 10. No, it depends on how you count the microcephalic fetuses, etc. So yes, causes nasty disease. Anyone who listens to TWIV knows that, in fact, one of the co-hosts, I think last week, had dengue fever. There are also lots of what they call sequelae. And so what happens after you've had the disease and all kinds of symptoms that sort of keep coming back again and again and again over years and years and years. And that is very, very poorly understood, but still causes a lot of problems in terms of economic outputs, quality of life, etc. So yeah, maybe not that many people die from these things, but there's still major issues in terms of disease. I want to briefly cover West Nile. Oh, I got the animation to work here too as well. Okay, so West Nile is really a bird disease, again, spread via mosquitoes, but there are what are called these dead-end hosts, either humans or horses, which then are getting sick, but they're not really contributing to the distribution of these diseases. So, and this happens back quite a lot, as you have these what are called the zoonotic diseases emerging from animal populations, coming into humans and causing quite severe disease phenotypes. Hantaviruses are a really nice example of that, where the humans get really, really sick, but there's practically no virus transmission from human to human. So watch out for those sneezes over there. Yeah? So the Zika virus, of course, this is what we're all excited about right now, except that Congress doesn't want to spend any money to study it. But large outbreaks in South and Central America, which may or may not have to do with antibody-mediated enhancement, which we'll get to in just a second here. Zika virus was, of course, originally found, I should say, here in Uganda and Central Africa, and it's been slowly moving eventually to the South Pacific, and then from there in a real explosion in South and Central America. here with all of these extra clinical case reports, and probably microcephaly, and the various different birth defects. Pretty clear correlations now. Whether there's an actual cause or not is still controversial, but people are moving further and further towards thinking there really is a causal effect with that. Quite why that is, again, may have to do with this antibody-mediated enhancement as well. But curiously enough, there's not very much known association of microcephaly in other parts of the world as opposed to what's happening in South and Central America right now. So important aspects, forget the disease stuff, of the flaviviruses is how they're replicating. They are enveloped viruses, come in through the endosome, low pH mediated conformational change, membrane fusion, all stuff we've talked about a whole bunch already. They are made as polyproteins. These are, again, enveloped viruses, and so the actual polyprotein itself is made on some of the membranes that are present inside the cell. These are all the non-structural proteins, all of the orange ones here. The structural proteins are the ones which are shown here in blue. Make RNA, again, it's a positive strand RNA virus. You've got to have a little bit of negative strand in order to make more of your positive strand. And it's the RNA-dependent RNA polymerases, which are sitting here at the very 3' end of your genome, which are actually making all of these copies of the RNA, which again gets translated mostly through iris-mediated processes. The other thing, which of course all of you are asking, what about the 3' end? 5' end, you've got irises. What about the 3' end? Exactly the question you're going to ask. I know, Trevor, that's why I beat you to it. One of the really interesting aspects about these flaviviruses is that in almost all cases, you have base pairing interactions that seem to happen between the 5' and 3' ends of the RNAs. And so all of these guys here should actually be shown as circles. And it's probably that base pairing interaction between the 5' and 3' end which is protecting the 3' end from getting degraded. And it turns out if you actually make changes in the sequence at the 3' end so that it doesn't base pair anymore to the 5' end, then the genomes are way less stable. So, yeah, really interesting aspect of how these things are being protected. I did want to emphasize this aspect of antibody-mediated enhancement. Clearly a big deal as far as dengue virus is concerned, may be part of the problem that we're having currently with Zika. So normally, flaviviruses here interact with the receptor. You have receptor-mediated endocytosis, comes inside the cell, changes, etc., replicate at these membranes. If, however, you have a small amount of antibodies around that will recognize the surface, but not completely surround the surface with antibodies, which would happen in a normal productive antibody response, what will happen is that these antibodies will bind to antibody receptors on a number of cells and then bring the virus inside those cells and potentially even cells that wouldn't have had the virus receptor on them in the first place. So what it's doing is extending the different kinds of cells that these viruses can now infect and giving you a much more advanced disease. This is very clearly shown for dengue, 4 different types of dengue. You have antibodies to one. They'll protect you very well to that one particular kind, but some of the other kinds, they'll actually cause this problem of antibody-mediated enhancement. So any questions about this? I think it's an important concept that I was really unclear on before. It has to do with the antibody receptors that are present on a number of cells. Okay, that's just this square thing here. Polyprotein, I think this is the only slide that Dr. Hirsch used that I gave him. We've got caps here down at the 5' end of the genome, non-structural proteins down here at the 3' end of the genome. Again, base pairing between the 3' and the 5' ends. Where does the cap come from? It's made by the viral proteins. So it's all viral proteins which are making these cap structures. and again made as a polyprotein, chopped into different pieces by not only the viral protease, but in this case also cellular proteases, which are involved here. And so I like this diagram here, where we've got viral proteases, it's all the green dots, host proteases, which are either the black stars or orange stars. So here, orange stars, black stars, this is all cellular proteases. And here, these are all the viral proteases that are chopping apart. Viral proteases mostly cutting apart your non-structural proteins, whereas the host proteins are mostly involved here in cutting the structural proteins. This one right here, PRM, this is the protease which takes place at the, or proteolytic cleavage, I should say, that takes place right at the very end of infection, right as you're putting those virus particles together. And it's that last cleavage that takes place which sort of primes the virus to now be infectious. So it's after everything else has come together. You've got your whole virion all ready to go, but it can't actually undergo binding to receptors until you have that last proteolysis event that takes place. This is also true for influenza viruses, for many of these other viruses as well. Okay, flaviviruses? Yes, no, indifferent? Good. What about these guys? What kind of virus are we talking about now? Coronaviruses, the SARS coronavirus, and lots and lots and lots of bat coronaviruses. Way more bat coronaviruses that are out there than there are coronaviruses that are infecting humans, at least in terms of the molecular diversity. And that was basically just what I wanted to emphasize here, is that these sequences, because there's no horizontal difference here in the tree, means these sequences are practically identical to each other, but they're also very similar, because of these short distances here, to bat SARS coronaviruses as well. The other thing to point out, again, when you're looking at these trees, anytime you see a number like 37, you could say this is probably crap. Anytime that you see a number like 90 or 100, it's actually really pretty reasonable. So that this group here, this group is a sister group to that bat coronavirus, yeah, maybe. That all of these bat coronaviruses are related to each other, definitely yes. So just say looking at trees and looking at some of these structures. Coronaviruses are, what was that again? Beluga whale? Yeah. The largest of the positive strands, actually of any of the RNA virus genomes. SW1 is that beluga virus, beluga whale virus. We're talking about over 30,000 bases in length, really ginormous genomes. They also have caps and poly-A tails. Here, instead of all being one polyprotein, they have one really big polyprotein, but then a whole set of their structural proteins now down here at the 3' end of your genome. Why the heck are you put your structural proteins down at the 3' end of your genome? Well, that's because it's made through lots of nested subgenomic RNAs. So messenger RNA production for the coronaviruses has to do with the RNA-dependent RNA polymerase, just like all RNA-dependent RNA polymerases, replicating from 5' to 3', which is going to be from 3' to 5' along a template. But these RNA-dependent RNA polymerases can jump, and they'll jump to this sequence right next to the 5' leader sequence that's present at the 5' end of the genome. What this will do is generate multiple subgenomic negative strands. We have lots and lots of copies of the sequence here, which corresponds to the very 3' end of your genome. These guys will get replicated again by the RNA-dependent RNA polymerase, generating multiple messenger RNAs here, which will make all of your structural proteins. Of course, you need way more of your structural proteins than you need of your genome, and so that's how you end up with all of these multiple copies. It's all about the nested subgenomic RNAs and the RNA polymerase being able to jump whenever it hits any of these repeated sequences here. So nested subgenomic RNAs. Amazing way that evolution has figured out how to make lots of structural proteins relative to everything else. MERS coronavirus in Korea, Middle East respiratory syndrome. The main thing here is one case in 2015, gone 3 months later. So really impressive process on how you can have outbreaks, but at least with good public health measures, get rid of them quite quickly. Whereas MERS is a reservoir, these cute little camel guys here, probably again, typical reservoir, not causing much disease as far as we can tell in all of these camels. Questions on positive strand RNA viruses before we talk about the negative strand ones. So now I'll go over to the other side. So vaccinate, vaccinate, vaccinate. All about measles. Measles was a really big deal in terms of childhood disease. We now have a really good vaccine against measles, and measles has almost completely been eliminated in the US, except for people who don't vaccinate, or for measles, which comes from parts of the world where there is a lot of endemic measles. And it's not just places like Ethiopia, Nigeria, but also England, France, and Germany, where we have a lot of the imported cases which are coming into the US. So vaccinate, vaccinate, vaccinate. How many more times do I need to say it? Good. So that's the vaccination aspect of things. And they talk about vaccines again. We talked about flu, if we get there, which we probably won't, but we just talked about it. The N proteins or NP proteins for paramyxos and rhabdos, but also for the filoviruses, the complete opposite of what we saw with TMV. Now those proteins are bound on the inside of the RNA so that the RNA can be replicated. So you can make messenger RNA from it, you can make genomes from it. And it's binding to the very ends, here at the very 3' end, but also the very 5' end of the genome. And these proteins, it's the protein binding here that seems to be protecting that RNA from the hungry exonucleases that it love to chew in at the ends of your genome. And that these RNA binding proteins not only protect, again, from the nasty exonucleases, but they also provide a structure because they're all helical. And what that means is the sequences which are right here at the end of your genome are right next to sequences which are here. And that means that the sequences here are just as important as the sequences here in terms of getting your transcription, replication, etc. to take place. We talked about how you make the individual messenger RNAs from paramyxo, rhabdo, and filoviruses. This is where you get stuttering, Peter. Where you have intergenic sequences, the L protein, which I was wondering where you get caps and tails from for all these messenger RNAs. It turns out it's the L protein that not only is the RNA-dependent RNA polymerase, but also the methylase and tailing enzyme. Amazing that this one protein does all these things. Very cool. So it'll bind to the 3' end of your genome. And again, it's that structure with the helical structure here. Binds there, starts to transcribe, makes messenger RNA for your nucleocapsid protein, gets this intergenic sequence, stutters, puts on a poly A tail. Some cases, the polymerase will fall off and your messenger RNA will be there. Other cases, the polymerase will stay on, reinitiate a new messenger RNA, get to the next intergenic sequence, etc., all the way down the rest of the genome. If, however, you've got lots of this protein encoded by this N gene around, or NP in the case of the filoviruses, that will bind to the genome, and when it's completely bound to your new genome sequence, what happens is you start replicating antigenome, which will then lead to genome sequence, and it's all about the concentration of the N protein, and particularly any unbound N proteins. If you've got extra N protein, that means you've replicated, you haven't replicated your genome yet. You've made enough of the protein so that you can make genome, and that genome can then end up being packaged. Presence of the N protein also stops stuttering. It stops this stopping-starting process as it's moving through. Filoviruses are just big rhabdoviruses for the most part, with a couple of differences. Those main differences being the VP30 protein here, which is our regulator of messenger RNA production, and the glycoprotein, which can also be formed as a secreted glycoprotein. Otherwise, extremely similar at the molecular level. Very different in terms of diseases that they cause. And I was going to go and look that up, but didn't have time to before class, is why you have such a big difference as far as these things are concerned. Slight difference here in terms of the stopping and starting. In some cases, in these filovirus genomes, you don't have a big separation like you do in the paramyxo and the rhabdos. These guys actually then overlap with each other. I mentioned, quote-unquote, transcriptional regulation. Again, it's about VP30. In the absence of VP30, you block messenger RNA formation. The presence of VP30 allows messenger RNA formation. Why am I beating on this so much? It's because one of the better vaccines out there for Ebola virus is one which is missing the VP30 protein. So you end up getting good antibodies, but you don't have to worry about these things replicating. What did we have with Ebola until 10 years ago? About 500 cases. What did we have last year? 30,000 cases, having to do with all kinds of different aspects about public health in West Africa, not dealing with the disease appropriately, and people got all freaked out about it for no apparent reason. So did want to mention really briefly the other vaccine for Ebola, which is the VSV. VSV is what kind of virus? A rhabdovirus, which now has the Zaire Ebola virus GP30, which is the, actually no, this would be the GP protein here. Get rid of that GP30XX here. So it's the missing its own glycoprotein and has the Ebola virus glycoprotein. One of the things that I wanted to mention here is that this has actually a lot to do with how you go about setting up vaccine trials. How do you go about setting up vaccine trials for a really nasty, deadly disease that you may actually be providing some protection to based on people then actually getting this vaccine? So it's really ethically questionable to be giving people a vaccine in a region where they're likely to die if they don't get the vaccine. And so what they did here was a really interesting study, although a ring study, which is where they had people who were infected. They would immunize people who were their contacts, and particularly medical personnel. But some they would give a vaccine to 6 months after other ones, and just basically see what was happening in that particular 6 months. But everyone got the vaccine. And so it looked as if it was working really quite well, but the numbers were so small that it was really hard to tell. But I thought this was really interesting. Usually with your classic kind of clinical trial, it's double-blind, et cetera. Nobody knows until you actually look at the data, and so you can actually analyze them. That's really questionable in terms of some of these really lethal diseases, and particularly in areas where informed consent may not be as informed as you might like it to be. So I just wanted to re-mention that trial. It's really interesting. If you're interested in looking at this really unique kind of trial, go and check out that website. I'm not going to get into a whole bunch of details on the exam other than the 8 questions that I wrote. No. So 3 more minutes. Flu. We've kind of talked a bunch about flu already. One thing to emphasize here, as is true for actually all of those other negative strand RNA viruses we've talked about, the vast majority of the proteins that are coded for in the genome are present in the virions. Of course, you have to have the RNA-dependent RNA polymerase there because as a negative strand genome, you've got to be able to make messenger RNA before anything else can happen there. And we've got our hemagglutinin, uraminidase, and then these channel proteins. Hemagglutinin, classic receptor binding protein, classic change in structure upon change in pH. classic in that it has this fusion peptide which is exposed by proteolysis by a cellular protease. That's at least the second time you've heard me say something like that today. So processes which are happening there. Messenger RNA production in these influenza viruses, the cap snatchers, the virus genome segments are all imported into the nucleus through some of the activity of the non-structural proteins. In the nucleus, the polymerase proteins, or the subunits thereof, PB1, PB2, and PA, will bind to caps of cellular RNAs, cut off the caps, and then use that cap as the primer to make the messenger RNA for each of the individual segments of the genome of the virus. And nice stuttering, Peter's favorite thing to have happen here, happens at the end of the genome with these poly-U's. What happens once you've made enough of your structural proteins? They start encapsulating these things, you don't steal caps, and you don't undergo stuttering anymore. Where do we stand this year? Relatively low flu year, most of it being H1N1 from the swine flu outbreak of 2009, and we've gone quite a long way down here, but it's a little late. Everyone's favorite number here should be this one right here, deaths per year in the USA, relative to number of deaths from Ebola. Okay, we'll leave it at that. Did want to mention pandemic flu again really briefly. These have been mostly due to reassortment. So the fact you've got all these different genome segments means that when you have infection of one particular cell with multiple different influenza viruses, those genome segments can mix and match. And that's what seems to be causing the problem for most of these outbreaks, with, again, the potential exception of the Spanish flu, which really actually seemed to have been a mostly avian flu, which came through there. We'll skip these preparations for pandemic flu. Again, we didn't really get to them at the end anyway. Good luck on the exam. Any more questions, feel free to email me or come ask me questions right now.